But I'm really excited tonight to present two speakers we have. Both of them are graduate scholarship recipients. The MMS has done graduate scholarships for, I'm going to say 10 years now, because I just looked back at the history and we gave them away in 2015. So if you count 2015 and 2014, it's actually been 10 different years. This year we gave away 4 graduate scholarships. And like I said, tonight we have our first 2 speakers, and I'm really excited to hear their presentations. Ryan Franke, some of you will remember him, and he may seem familiar to you. He did our newsletter for a while. He used to help out with forays. He's been really active in our club, and now is doing a lot of research and has a new little one at home. So like always, life gets busy, but he's going to present tonight biological control of invasive buckthorn with native fungi. And Ryan sent me a nice little bio. He grew up in Eagan, Minnesota, and learned to revere the natural world at a very young age. During a fall camping trip along Lake Superior in 2015, he became fascinated with fungi and shortly thereafter joined MMS. So his fascination with fungi mushroomed after joining the society, and he opened a gourmet mushroom cultivation business in 2017. Like I said, he was the editor of the MMS newsletter from 2018 to 2020, and most recently joined the mycology-focused Forest Pathology Lab at the U of M in 2023. So thank you, Ryan, for being here tonight. Thank you.
Yeah, thanks for the introduction, Kathy. Really happy to be presenting to the society tonight, as I've been a longstanding member, more absent in the recent years because life has gotten so busy, but here I am back. And my research is looking at developing a fungal biological control for fungi. And so the presentation tonight is titled Fungi Associated with Dead and Dying Invasive Buckthorn as Potential Agents of Biological Control for Invasive Buckthorn. And I'm working on this project in my professor and principal investigator's lab, Robert Blanchette, here at the University of Minnesota. And also working on the project with us is senior research scientist Ben Held in the Blanchette lab. And our research is funded by the Minnesota Invasive Terrestrial Plants and Pests Center, which in turn receives funding from the Minnesota State Lottery. So if you've ever bought a lottery ticket in the past few years, you may have helped support this research, which is kind of fun.
Right. If I may step in for a second, Ryan, that gives us a point, is that the lottery constitutional amendment is up for renewal this year. So please remember to vote this fall to keep that funding intact.
Absolutely. Yeah, you're absolutely right. We want to keep funding the environmental initiatives through the lottery. So that's, I think it's like, if it's voted yes, it's maybe for another 50 years that will fund the environmental initiatives through the lottery. So it's a big impact vote. So the problem that we're addressing in this research is buckthorn, which is introduced species from Europe, both glossy buckthorn and common buckthorn. And these species, as you probably know, being a Minnesota resident, outcrowd native species in different habitats, and especially in disturbed areas. And these invasive buckthorns are very efficient at photosynthesizing, and essentially they're able to arrest the process of forest succession. They grow up underneath the aspen trees, which are the first pioneers in transitioning from a prairie to a forest system. And in place of the maples and oaks and other slower growing trees that would normally grow up underneath the aspens. And essentially, we're not seeing the continuation of forest succession in a lot of places in our state because of this plant. Not to mention just herbaceous native understory plants are also being outcrowded. They can't survive underneath the low light levels underneath a canopy of buckthorn. And so this is a big problem in our state. And so an overview of our project, the kind of the steps for developing a mycoherbicide. We've begun with collecting fungi from dead and dying buckthorn in hopes of isolating fungi that are responsible for the disease in these dead and dying buckthorn. And we've done a lot of this collection last summer. So we got a check mark next to that bullet point there. Now we plan to test these fungi for their ability to cause disease on healthy buckthorn. So just because we isolated these fungi from dead and dying buckthorn doesn't mean that they're responsible for the disease. So we need to test their ability for pathogenesis on healthy buckthorn. And we're in the process of testing our collection of fungi currently. And lastly, we will be developing a methodology for application of the most pathogenic fungi on buckthorn at a field scale. And so similar to how herbicides are applied to invasive plant species, we plan to use some of the existing technologies out there, like this, perhaps just for example, this green shoots herbicide applicator to apply a fungal inoculum instead of a chemical herbicide. So developing that methodology is kind of the last step in developing a mycoherbicide for land managers to use to control buckthorn. And so last summer, we visited 17 sites across Minnesota and Wisconsin with dead and dying buckthorn. And most of these sites were brought to our attention by various collaborators, particularly the Minnesota DNR, the Minnesota Department of Agriculture, the nonprofit Friends of the Mississippi River, a lot of different citizen scientists who sent myself or my PI, Bob, an email telling us that they had dead and dying buckthorn in their backyards, and also the Wisconsin Department of Natural Resources for our sites in Wisconsin. And so this is a large collaboration with various private and state entities and our research group to collect fungi from dead and dying buckthorn. And so when we visited these sites, we saw a variety of symptoms. The photo on the left is myself holding a dead buckthorn stem. And you can see there's a couple of little sprouts at the bottom of the stem. And those plants we sampled because we preferentially sampled because having a little bit of living tissue left meant that there was still a niche for a disease-causing organism to exist on the plant, as opposed to a buckthorn that had been killed for 6 months and was completely dead. The organism that caused the disease would likely have been outcompeted by a decomposing fungus or suite of fungi. And so those are kind of what we looked for in our sample locations. And some of the symptoms on the stems included these cankers in the center photo. So a canker is essentially a portion of cambium tissue, inner bark, vascular tissue that has died. And you can see in the center of that stem, looks kind of like rotten and white, and that's basically some cankered tissue, and it's elongate, and the tree has responded by forming a ring of callous, thick tissue around that canker to localize it to one portion of the stem and to keep it from girdling and killing the tree. So we saw some of these localized cankers. In the photo on the top right, there's cut face of a diffuse canker. You can see the healthy cambium vascular tissue is kind of a light yellow color on the bottom portion of the stem, and on the top portion of the circumference of that stem is blackened, and that has been killed tissue. And you can see the tree has not responded by forming a ring of callus tissue to kind of wall off that canker. And so we isolated fungi from that tree. And on the bottom right photo, you can see this buckthorn tree had multiple canker infections that had killed cambium tissue at various spots around the circumference of that stem. And we also saw signs of fungi at these various locations. And for example, the photo on the left is a Urpex lacteus fruiting body. So this is a basidiomycete fungus, and it forms a milky white mushroom with teeth. And this is a basidiomycete fungus that causes canker rot on limbs of trees where it has infected. And this is a kind of a generalist pathogen, and you may have seen this fungus as you've been out foraying in the woods, covering the limbs of branches that it has infected and killed. And so we saw this Urpex fruiting bodies a few occasions at different sites last summer. And then the photo in the middle, there's some little white pustules poking through the outer bark there, and on the photo on the right, there's some pinkish pustules that's exuding through the bark, and these are likely belong to ascomycete fungi, these microfungi. And so these are the fruiting bodies. They're extremely small, and you likely will not notice them if you're out looking for chanterelles and morels. However, if you're looking for them, you'll start seeing them everywhere. The ascomycetes are a very successful and common group of fungi, and so we also were isolating from these ascomycete fruiting bodies that we were seeing at these locations with dead and dying buckthorn. So we used hand tools, saws, and pickaxes to sample above ground and below ground disease tissue, both common buckthorn and glossy buckthorn, depending on which was present at the site. And then we brought our samples back to the lab and isolated from the diseased tissue. And so the photo on the left, there's a large localized canker on glossy buckthorn. And the photo in the center, there's a diffuse canker running up and down the stem on a common buckthorn. And you can see common buckthorn has a distinct orange colored cambium tissue. And so on the right portion of that stem is healthy cambium. And you can see it's in contrast with the blackened cambium tissue on the left. And where I sampled from the stem was right at the interaction zone between healthy and dead tissue, because that is the most likely place to isolate the fungus causing disease. And the photo on the right depicts common buckthorn with root rot. And so we also isolated from some root rot at a couple of locations. And so after excising a piece of diseased tissue, I placed the tissue onto one of 3 different selective media types. And basically, we used selective media in order to capture not only the fast growing fungi, but also the slower growing fungi that would be outcompeted by faster growing fungi. And so through using 3 different types of media, we were able to collect a range of faster growing to slower growing fungi, different groups of fungi. And after the hyphae grew out from the piece of plant tissue, I would subculture a small piece of hyphae into a new petri dish, and in that way I obtained pure cultures of fungi from dead and dying buckthorn. And so, let's see. And so, in all, we isolated 331 fungi from dead and dying buckthorn, and you can see in the photos here that there's quite a diversity just based on kind of macro morphology, all sorts of different colors and textures and growth rates. So even before sequencing, it was clear that there wasn't just a couple dozen fungi associated with dead and dying buckthorn, but probably at least a few dozen. And so far, we've used DNA sequencing to identify 294 of our isolates as 89 unique species belonging to 64 genera. And so in case, genera and isolates is kind of confusing terminology for you, I'm going to do just a real quick basic review of taxonomical ranks in biology. So in this example, we have morels, which were not found on dead and dying buckthorn, but it's a common mushroom that most people know, so I'm using it for this example. And wouldn't it be great if we did find them on dead and dying buckthorn, though? And so we have the black morel in the photo on the left and the yellow morel in the photo on the right. Both are in the genus Morchella. And the second word in their name is their species. So we have the black morel is Morchella elata, and the yellow morel is Morchella esculenta. And because they're in the same genus, that basically means that they're closely related to one another. If you look at the table on the left, one level above genus is family, the Morchellaceae, and above that is the order, Pezizales. Above that is class. Above class is phylum, Ascomycota, and above phylum is kingdom fungi. And so when we talk about having 89 unique species belonging to 64 genera, you can kind of think of it like having 64 groups, and within those 64 groups, there's 89 kind of like subgroups. And so this illustration shows the genera that we collected across all 17 sites in proportion to their relative abundance. And so the larger boxes denote genera for which we collected many isolates. And the smaller boxes on the right of the illustration depict genera for which we collected just a few isolates. And actually all of the kind of square dimension boxes, all the small squares on the bottom right corner of the illustration represent 32 genera for which we just had one isolate each. And on the other side of the spectrum, we had 6 genera with more than 14 isolates. And those are these 6 big boxes on the left. And we collected 26 genera for which we had somewhere between 14 and 1 isolate in the genus. And so what this is kind of showing is that there's a fairly species-rich assembly of fungi that is associated with dead and dying buckthorn. It's not just a few characters that are causing disease, but there's quite a few fungi there. And so we broke down the fungal genera by their predominant lifestyle as referenced in the scientific literature. And so what we see here on the y-axis is the number of genera. And on the x-axis, we have the lifestyle that characterizes the genus. And so the most abundant, abundant lifestyles for the genera that we isolated were the canker pathogenic lifestyles and the saprophytic lifestyle. And this is not really too surprising because we were isolating from diseased tissue, hence the pathogens. And shortly after a pathogen moves through some tissue, the tissue is dead and is now available for saprophytic fungi to exploit the new niche, the resources. And so, saprophytic fungi in this category include ubiquitous fungi like penicillium, molds, and alternaria. And the saprophytes and the canker pathogens were followed by wood decay fungi, which is also not really too surprising, being that the trees were recently killed. There is an abundance of wood for wood decay fungi to exploit. And followed by the wood decay fungi, we isolated 4 genera that can be characterized as endophytes, which basically means that these fungi inhabit plants without causing outward symptoms of disease. And then we also isolated 4 genera that can be characterized as entomopathogens, followed by foliar pathogens, followed by 1 genus that is mycoparasite. And then there is quite a few genera that I could not place into one of these categories based on the available scientific literature. So there's just not really enough known about these fungi to be able to place them into a lifestyle. And so what we're interested in are the pathogens, because we want to kill buckthorn. And so we collected 29 different pathogenic species belonging to 14 genera, which is great. It's like it's a full tool belt. And so I'm not going to go too in depth into this table, but essentially this table is all the pathogenic fungi organized by genus that we collected from dead and dying buckthorn. And it's organized in descending order of relative abundance of the genus. And so if we look at the top row, we have the Fusarium genus, which represented 8.69% of our total isolates belong to this genus. And we collected Fusarium at 9 of the total 17 sites. And the species that belong to the genus include the Fusarium acuminatum, Fusarium paeonii, Fusarium avicennium, Fusarium juglandicola, Fusarium lateritium, Fusarium quercinum, Fusarium sambucinum, and Fusarium solani, and so on and so forth down the table. Something to note about this table is that all but 2 genera belong to the Ascomycota phylum. And so these are the microfungi that don't produce, by and large, with some exceptions, don't produce large, fresh, fleshy fruit bodies. And so the 2 Basidiomycota genera that we isolated were Urpex, which I mentioned earlier in the presentation as forming that milky white tooth fruiting bodies, and also Hymenocete tenuis, which is a canker rot fungi that can is a kind of a general pathogen of woody plants. And so we're excited to isolate that fungus in addition to the others in the table. In addition to fungi isolated from dead and dying buckthorn, we also isolated some fungi from non-buckthorn hosts. And we did this because we knew that these fungi are generalist pathogens and may be able to kill buckthorn. And you can see that all of these isolations were made in the state of Minnesota. So even though they are pathogens, they are native and common across the state. And so using them as a biological control isn't necessarily isn't introducing a new pathogen to the region and isn't introducing a new threat. However, it's just kind of applying it in a targeted way to buckthorn. And so the fungi that we isolated from other hosts include fungi in the Hymenocataceae family, which is a canker rot family of fungi. We have a few isolates of Serena unicolor, which is another canker rot fungus. You can see this fungus fruiting, all over the Twin Cities area on stressed trees. And Verticillium dahliae, we have a couple of isolates, which is a wilt-causing fungus. And we also have a few isolates of Chondrosterium purpureum, which we were really excited to find and work with because this fungus is being used currently in a registered commercial product in Canada as a mycoherbicide applied in managed timber stands to cut stumps of weed trees. So that way the weed trees don't grow back when they're managing timber stands. And this is being used as an alternative to chemical herbicides, reducing environmental consequences. And so we know that this fungus is being used successfully on other hardwoods commercially. And so we're excited to find this in the state. And when I first started on the project last summer, I was talking with my PI, Bob, and he was saying, I don't know if this fungus is in Minnesota. I think it could be somewhere up in the northeast portion of the state, but I'm not positive. And so the first thing I did was I hopped on iNaturalist and I looked for observations of the fungus. And sure enough, there was a couple of observations in the Twin Cities area. And one of those was made by MMS member that most of you probably know, John Lampert. And this is the fungus here growing on John's son's tree in Lakeville. And John sent me a photo. I reached out to John, said, hey, could I come and take a look at this fungus? And John said, sure, it's growing on my son's tree. Come on down, take a look and grab a sample. And so I went down there and isolated from the decaying wood behind the fruiting bodies and sequenced the DNA, and it came back as Chondrosterium purpureum. And we were pretty excited to have an isolate that we could work with. And since then, I've found an additional 3 isolates of Chondrosterium purpureum. And this fungus may be more commonplace in the state than we had thought, or that was been documented in online. And it's kind of an inconspicuous fungus. It's beautiful when there's high humidity and it's just rained and it's got fresh fruiting bodies, like in the photo that John took here. But most of the year it looks pretty drab and it's easy to overlook. But this may be a pretty common fungus, and I imagine I'll start seeing it everywhere now. So last fall, we took 12 of our isolates that we thought were the most likely candidates for being able to kill buckthorn. And we inoculated them into 12 trees per isolate in a field inoculation experiment. And we conducted this experiment at 2 replicate sites in the Twin Cities area. And we'll be harvesting the inoculated stems this summer. And we'll be measuring the size of the resulting canker around the inoculation point as an indicator for pathogenicity on buckthorn. So the larger the canker, the more pathogenic the fungus is on buckthorn. And so how we did this was we grew the isolates out on wood chips, as well as on barley grains. So we had 2 different types of inoculum per isolate. And we made a wound in each buckthorn tree and then inserted the inoculum into the wound. And then we wrapped the inoculation site with parafilm wax paper to prevent the fungus from desiccating. And then we wrapped the parafilm with duct tape to protect the fungus from UV radiation. And then we crossed our fingers. This summer, we will be sampling fungi from additional sites in Minnesota and Wisconsin with dead and dying buckthorn. And this is a call out to all you citizen scientists to report locations to us with dead and dying buckthorn. There is a Google form, which makes it really easy to do so. The link for the form is shown here on the slide. So if you want to take a minute to scrawl the link out on a scratch piece of paper or post-it note, feel free to do that. Or you can reach out to me at my email address, and I can send you the form that way. But we are looking for more locations with dead and dying buckthorn. And we will also, this spring, we're replicating the fall 2023 field inoculation experiment that I just highlighted in the previous few slides. And we'll be basically replicating the experiment in the springtime to test for an effect of season of inoculation on the degree of pathogenicity for the isolates. So do the fungi cause a greater degree of disease when inoculated in the springtime into buckthorn versus the fall time, or vice versa? This summer, we'll be harvesting the buckthorn stems from the fall field experiment. And this fall, we'll be harvesting the buckthorn stems from the spring field experiment. This summer, we'll also be optimizing inoculum viability and pathogenicity. And so we'll be using our most pathogenic isolates as evaluated by the 2023 and 2024 field experiments, and creating various formulations for application onto cut stumps of buckthorn. And so you can imagine land managers going through and cutting their buckthorn and then applying an inoculum, fungal inoculum to the cut stump. And so we'll be testing different media formulations to be applied to cut stumps, and also testing different devices for applying those formulations. So we're looking for both efficiency and efficacy of biological control. And I want to acknowledge here at the end of my presentation our field collaborators, the Minnesota Department of Natural Resources, the Minnesota Department of Ag, Wisconsin Department of Natural Resources, Friends of the Mississippi River, 3M, which is hosting one of our field inoculation sites, Flint Hills Resources, which is also hosting one of our inoculation field sites, a whole host of citizen scientists that have brought to our attention locations with dead and dying buckthorn, and my comrades in the Forest Pathology Lab here at the U, fellow graduate students, Nick Raytar, Andrew Mann, and Colin Peters. And last but not least, my wife, Alicia, and our 21-month-old son, Malachi, who paradoxically is able to humble and inspire me at the same time. And I hope we have some time for some questions.
Yeah, thank you so much. That was amazing. We already do have a lot of questions. And I wanted to first off thank Nick for putting, I think he put your Google form in the chat. So there's a link to that. And then there's a question from Paul Egger. Actually, Paul, you're welcome to unmute as well. It says, you mentioned that there's a fungus being used to control weed trees. And can you talk more about it? And does it or might it work on buckthorn?
Yeah, so Chondrosterium purpureum, see this fungus back here, is currently registered as a commercial product in Canada and as a mycoherbicide. And it's being applied by land managers in managed timber stands. And basically, in a managed timber stand, different trees that are not the tree that's being harvested for timber. So let's say like birch trees or alder trees or maple trees, that are not, the pine trees that are being grown for timber are, when they're volunteer themselves in these managed timbered fields or plots, they're taking resources away from the timber trees, and water and nutrients. And so it's a common commercial practice to go in and cut these trees out of managed timber stands. And however, a lot of these trees are able to vigorously resprout from the base of a cut stump. And the practice for many decades has been to apply an herbicide like glyphosate or some other herbicide to the cut stump to prevent resprouting, to basically kill the roots below beneath the cut stump. However, there's I think it was in the early 2000s, this product came out that's Chondrosterium purpureum, and it's I'm not sure about how widely it's used in the industry, but I do know it's a commercial product that's being produced and being used at least at some scale across the country. Unfortunately, and kind of unfortunately, just because it's being used in Canada doesn't mean it can be used here, because prior to our research, it wasn't validated that this fungus is resident in the state of Minnesota. And so we're doing our own kind of research to validate the efficacy of this fungus and the ability of this fungus to serve as a mycoherbicide in our state.
Thank you. There's a bunch of other questions, but I'm going to jump to this one at the bottom because it seems really related to that. From Alicia Waters, it says, will you be looking at the effects on surrounding native vegetation after applying the fungi to kill buckthorn? She says, I'm curious about the residual effects once the buckthorn is dead. And I think you're sort of hinting at that with the Chondrosterium purpureum, that it's not necessarily allowed here because the effects maybe haven't been studied yet on other things.
Yeah, so I guess there's a couple of points I'd like to illustrate. One is that there was a study in 2005 on the ecological consequences of using Chondrosterium purpureum as a targeted mycoherbicide. And basically what these researchers found was that there was no increase in incidence of disease caused by Chondrosterium purpureum in the area where it was used as a mycoherbicide. And so that's suggests that because it was native to the area, the trees that it's co-evolved with for millennia have some kind of level of resistance to the fungus, and so it's not going to cause an epidemic. And we speculate that because this fungus is also native to what we're finding this fungus here in the Twin Cities, and it's not causing epidemics. It's actually kind of hard to find, and so because it's already here, increasing it in its numbers in targeted locations likely won't result in epidemics. Yeah.
And then how about, so the fungi that you're identifying, the isolates, those are all native as well. So you're thinking that there's ideally not residual effects from the ones you're putting in the buckthorn now. Is that true?
That's right. Yeah, because all the fungi are resident to Minnesota, they're already here. So this is a kind of subfield of biological control called augmentative biocontrol, where you're basically taking a organism that's already present in the landscape and increasing its numbers in one particular target So in this case, it'd be a bunch of spores on the cut face of a buckthorn. And basically you're kind of you're giving a huge leg up to the pathogen by cutting the buckthorn and then applying a bunch of spores to the stump, and then putting in some kind of formulation that protects it from desiccation and UV radiation. And so it's kind of like chopping someone's arm off and putting a bunch of staph bacteria on their arm or something like that. Where staph bacteria is present, it's all over the place, but it's not causing epidemics because we're not walking around with amputated limbs, as kind of an analogy. And this is in contrast with classical biocontrol, which isn't as hot of a field anymore as it used to be, because there were some unintended ecological consequences. And basically what was done was pathogens or predators of an invasive species were found in their native range. So in this case, it'd be in Eurasia, where buckthorn is native, and those would be brought across to the invasive range of the invasive species and introduced. And basically what this did was introduce non-native species. And sometimes these species introduced to control the invasive species became invasive themselves. And so this is not what we're doing, because it's dangerous and has led to epidemics and endangering native species in a lot of different instances. But again, this is not what we're doing.
Thank you. I'm going to pop back to Ron's question. He's asking, what about Puccinia rust on buckthorn? Does it kill buckthorn?
Yeah, so the Puccinia oak crown rust is being studied by another lab here at the University of Minnesota, and it had been studied for, gosh, I don't know, 4 years or so, 4 or 5 years, by a graduate student, Nick Greetens, who recently graduated. And his kind of conclusions were that although this fungus does readily infect buckthorn and kind of decrease its fitness, it doesn't cause mortality. And essentially, he doesn't think, according to his research, that it's pathogenic enough to be able to kind of justify the costs of growing it and inoculating buckthorn with it, and that it's not a good option. Now, that being said, there's someone else in that lab who is now working on that project, and I think she's looking at collecting more isolates of oak crown rust and working with those. But yeah.
Awesome. And Barry's saying, I've seen where they put black plastic on buckthorn stumps to stop them regrowing. And do you know if that works?
I've tried that at my parents' house, and I loaded up the black plastic with wood chips to retain the black plastic in place. And I think it worked about 50% of the time, but in the other 50% of individuals, the stump sprouts poked right through the black plastic, and the tree continued photosynthesizing and growing. And so maybe thicker black plastic, maybe you get the really thick stuff. But yeah, I think it is possible. It's just you might have to be diligent and replace the black plastic every so often, or go out and trim the stump sprouts that poke through the black plastic and replace the plastic or something like that. But it's labor intensive. You have to kind of maintain it. And our hopes with the mycoherbicide is it's something that you apply once, just like a chemical herbicide, and you walk away and it kills the tree.
Perfect. Yeah. Judy Olson, I put tin cans over buckthorn that have been cut off, and it seems to stop them growing. But yeah, it seems like what you guys are testing too. I mean, we're all trying everything we can with buckthorn, but you guys are looking for something where you could maybe treat hundreds or thousands of acres. My question was maybe it's super obvious, but it was about the picture you showed that was called the hypo hatchet tree injector. Is
Oh,
that
yeah.
like it's like a sharp thing that holds spores that you can kind of introduce the spores with?
Yeah, it's a kind of a ingenious device that has a tube that flows through the bottom of the handle. And it basically, you connect that tube up to a source of, um, and how it's used currently is herbicide. And when the blade of the hatchet strikes impact against a tree, there's a mechanism that squirts and injects a little bit of that herbicide into the vascular tissue of the plant. And so it's yeah, it's kind of a neat thing. I think this device would make the most would provide the most utility for using like a wilt fungus, like a verticillium, where you just need to insert, a small bit of spores into the vascular tissue, and then the spores can travel throughout the vascular tissue and plug up the tissue and kill the plant. Um,
So maybe not like I could get one to make my shiitake inoculation faster.
Yeah, if you do, let me know how it goes.
And then Joan is asking, and I think Joan, you're asking about testing the fungi after animal foraging, like after having goats come in and eat everything down. And Joan, you're welcome to unmute if you want to ask your question in person.
Yeah, that's one part of the question. Also, whether or not after application, if there would be any animal implications as well. So kind of both sides of that.
Okay, sure. So you're wondering if like if we've been sampling or intend to sample buckthorn plants that have been browsed by goats in hopes of finding some fungi that are attracted to stressed buckthorn plants. Is that your question?
Correct. Yep, exactly.
Yeah. So we have not sampled from goat browse buckthorn, but we did sample from this buckthorn that was being experimentally managed with this new technique called critical cutting period. And this is being experimented with by the nonprofit Friends of the Mississippi River along a floodplain along the Mississippi River. And basically what they're doing is they're cutting the buckthorn off at about hip height in the early summer or late spring of the year. And then the buckthorn resprouts and they come back a month later and cut off all the resprouts. And then the buckthorn will resprout again. And they come back in the fall and cut off the sprouts again. And then the following year, if they revisit the site in the spring, and if it resprouts again, they again cut off the sprouts. And they're essentially taking the chainsaw and kind of just mauling the stumps of these buckthorn to cut off the sprouts. So they're like really stressed out and getting wounded really, really badly. And we thought it would be a great idea to go out and sample the fungi that are invading those plants that are being stressed out. And we collected a handful of isolates from those trees that were being treated with the critical period cutting. And yeah, I think it's a good idea to go out and sample buckthorn that's being stressed out by browsing. There's likely some fungi that are there that are attacking the plants.
Awesome.
Oh, and then your part two was like, could there be some implications for animals browsing on stumps that have been treated with a mycoherbicide?
Yes. Mhm.
Um, I I don't know for sure, but I know not many animals um, um, like persist or have like a large portion of their diet um, consist of like crust fungi, which, you know, a lot of these fungi that we're experimenting with are kind of crusty, um, you know, not very voluptuous fungi. Not many animals, you know, eat a lot of that. And so I don't imagine they'd be greatly, you know, attracted to browsing or eating these the fruiting bodies um, of these fungi. And you know, that's in contrast. What I first thought of was like um, uh, different uh, uh, ergo species, the fungus ergo, which is like an endophyte and a pathogen of grasses, and it you know, creates these alkaloid compounds that are toxic to livestock when the livestock grazes on them and can be a problem. But that's different because the livestock, you know, eat a lot of grass, and it's primarily what their diet consists of. And so I think I don't think that's a very good analogy, and I don't think that would be the case.
It's almost like you've taken away all the tasty bits of the buckthorn.
Okay. Mm.
Sorry.
No, you're right, Kathy. Yeah, thanks, Ryan.
Yeah, absolutely. Good question.
Awesome. Yeah, thank you so much, Ryan. That was amazing. I know that everyone's so concerned with buckthorn, and it's fun to see how just the deep science of some of the stuff we're interested in might be a solution that's already there. Like you said, it's like these are all native species, and just to isolate them and find them is really fascinating. So thank you so much.
Yeah, you're very welcome. Yeah, it's fun to present. Thanks for the great questions, everybody. And again, please use that Google form this summer, because we'll be collecting more fungi. And the more fungi we have, the better. You know, the fungus that solves the buckthorn problem could come from your backyard. So thanks in advance for using the form.
Thank you. Thank you. And now I want to introduce Kathleen Thompson. Kathleen is presenting mycorrhizal interactions and their connections to current and future forest structure. Kathleen is a fungal ecologist who was fascinated by fungal symbioses. Very recently, in fact, she said today, she just came from the graduation ceremony in Madison, Wisconsin, and drove here. So she just got into town, completed her PhD in botany at the University of Wisconsin-Madison, studying mycorrhizal interactions. And she previously completed a master's degree in ecology and evolutionary biology at Iowa State University, studying lichen diversity and ecology. She's led numerous workshops related to fungal biology and served on the Madison Mycological Society board for over 4 years. And when she's not musing over her fondness for fungi, you can find her tending to her sweet pups, Phoebe, Starbucks, and Corbin.
Okay, well, first of all, thank you all so much for having me. It's a delight to be here. Especially, yes, I just came in a few hours ago. My car is still absolutely loaded down with all of my boxes. So I'm excited to finally see my new apartment for the first time later tonight. So that'll be exciting. Yeah, I'll go ahead and dive right in. Actually, Kathy, before I get started, what is your usual set end time, just so I can keep that in mind in terms of my presentation as well? Oh, can you all hear me?
Sorry, we
that's okay.
tried to be done with everything and log off by 9-ish.
Okay, good to know. Just wanted to be sure. All right. Well, thanks again, all. I'm excited to share a little bit of what I just recently presented for my dissertation exit seminar here as part of my PhD work. And so yes, overall, what I've been doing for the last many years at the University of Wisconsin-Madison is focused on disentangling the context dependency of mycorrhizal relationships and doing this in the context of our forests. So there are two primary ways in which plants access nutrients. One of those is through their own roots, through their own plant roots, and the other is through close associations with other organisms, such as mycorrhizal fungi, in exchange for carbon. And if we look at the word mycorrhizal, myco means fungus, as many of you in this organization already probably recognize, and rhizal means root. So therefore, what we're talking about here are close associations between fungi and plant roots. And although mycorrhizal relationships are very common, they're quite poorly understood in terms of, given how ubiquitous and important we know that they are. And part of this muddling is because they're very context dependent. So they can act very differently in different conditions. And although there are multiple types or guilds, you might hear me use that word guilds of mycorrhizal fungi, ectomycorrhizal fungi, or EM, what I have pictured on the left here, and our buscular mycorrhizal fungi, or AM fungi, are the most abundant and widespread, and they constitute the vast majority of mycorrhizal partnerships with tree species. So ectomycorrhizal fungi tend to be the sort of fleshy fruiting body. So if you're thinking guild, agaric mushrooms, your classic mushrooms, yeah, that's what we're going to be talking about, ectomycorrhizal fungi. Our buscular mycorrhizal fungi, as I'll highlight here in a bit, tend to persist mostly underground. They don't produce above ground fruiting bodies for the most part. And so oftentimes you only see them with a microscope. So although both ectomycorrhizal fungi and our buscular mycorrhizal fungi fill this kind of general role as mycorrhizal symbionts, where they're trading services such as nutrients for carbon from plants, they're quite different when we start to consider morphology, or their body form, as well as physiology, how they function, as well as their evolutionary histories. And therefore, they have these very distinct partnerships with their plant hosts. For example, ectomycorrhizal fungi have a Hardeg's net, which is, if you look at the kind of larger subset picture there, it's essentially hyphae that's permeating through all the apoplastic space between plant cells. You can kind of see that green network of cells in between the plant cells in the diagram. And they also have this rather thick or thin, depending on what species, mantle along the outside. And I tend to think of that as the little tube socks of ectomycorrhizal fungi. It's this kind of green ring, if you can see my pointer here on the slide as well. Our buscular mycorrhizal fungi, on the other hand, tend to have a lot less biomass at the root zone. So they tend to make these little arbuscules, which sort of push into the plant cell membrane, at least, and create a lot of surface area in the cell. They have vesicles, but overall they have a lot less hyphae and biomass at the root zone. So very different. Maybe my computer, there we go, unfrozen. Another major distinction between these two groups is their differential ability to access nutrients from organic matter in soil. So ectomycorrhizal fungi possess a larger sort of collection or suite of saprobic enzymes than our buscular mycorrhizal fungi. And it gives them this really unique ability to break down complex molecules and what they call mine for nitrogen from organic matter. So that can be things such as organic molecules in the soil, leaf litter, that type of stuff. AM fungi instead tend to capitalize more on the effects of freely living saprotrophic bacteria and fungi, and basically scavenge for freely available mineral nitrogen instead. And so with this unique ability to mine for nutrients from organic matter, ectomycorrhizal fungi could offer an advantage to plants in nitrogen-limited environments, or more accurately, in areas where nutrients are bound up in organic matter, such as in boreal and temperate forests, where there's nitrogen present, but it's often in compounds and molecules that are hard to access. It's not like a freely available mineral fertilizer. However, it's also been hypothesized that maintaining an ectomycorrhizal symbiosis is more carbon costly to plant hosts. And if my computer will There we go. Poor computer's on its last leg after a dissertation here. So it's more carbon costly to the plant host, or has been hypothesized to be. So therefore, the context dependency of a given mycorrhizal interaction is also the result of which fungal partners we're talking about. And this all becomes really interesting when we start to consider forest structure. And when I say forest structure, what I mean is like, which trees are there, what species, what functional groups they are, how abundant are they? In other words, as you know, even driving from Madison over to Minnesota, as I did just earlier today, you can see that the forests change, even as you get just that little bit north. If you were to take a car and you were to drive from the tip of Florida all the way up to the tip of Maine, you're going to see a lot of forest changes along that route. So what drives that, essentially, is what I'm interested in. So currently, along that note, there's a distinct latitudinal gradient with regards to mycorrhizal associations in forested systems. So as we tend to be in these more northern latitudes, we tend to see a lot of trees that are associated with ectomycorrhizal fungi. Think pines, pretty much any coniferous tree. Think even oaks. Yeah, those are couple good examples here. But when we tend to be down more towards tropical latitudes, we tend to find plants and trees that are associated with arbuscular mycorrhizal fungi. And in temperate regions, it tends to be more site-specific and is sort of 50-50. And this pattern is really closely correlated with latitudinal patterns of soil organic matter, as well as nutrient limitation. So northern latitudes tend to have large pools of soil organic matter, so lots of organic material in the soil, but tend to have nitrogen limitation because their nitrogen is all bound up. Down in tropical latitudes, we tend to see smaller pools of soil organic matter and phosphorus limitation. So we have this kind of distinction going on here. And these parallels are also tied to differences in litter quality, as well as decomposition rates, decomposition rates as well as litter quality. So for example, ectomycorrhizal associated trees tend to have low quality litter. And when I say that, what I mean is like, it doesn't decompose very well. There's not a lot of nutrients, there's a lot of carbon. So you have to work really hard to get the nutrients that are in those leaves. And it's tough to break these down. And so therefore those litters tend to decompose slowly. And that's further exacerbated by the climate in these areas as well. You have a relatively short window for decomposition. And so when you have tough litter and a short window for that, you get slow decomposition overall. And that overall can lead to these large pools of soil organic matter and nitrogen limitation that we see, potentially. So given that, it's possible that why we see a lot of ectomycorrhizal trees in these areas is because they're needing fungi that have these enzymatic abilities to mine for the nitrogen that's there, that's bound up in these molecules. So it selects for trees that associate with ectomycorrhizal fungi. And when you have that, you tend to have trees with lower quality litter, which lead to these potential feedback loops in these areas, which is why we don't, haven't historically seen a lot of changes in these areas. Conversely, in tropical latitudes, sorry, my computer is running a little slow. Oh, here we go. Oh, but as I said, those are also hypothesized to come at an increased carbon cost. At tropical latitudes, however, arbuscular mycorrhizal associated trees tend to have higher quality litter, which paired with their increased climatic window leads to faster decomposition. Overall, then, we see smaller pools of soil organic matter, phosphorus limitation instead, which may select for arbuscular mycorrhizal fungi, because why pay the high carbon costs of ectomycorrhizal fungi when those services aren't necessarily needed? However, the specific factors driving these feedbacks aren't super well understood. So this is sort of a theoretical model thinking like, maybe this is why these systems have persisted the way that they are for so long, but we're also not sure how resilient they are to change. And that's an issue as we continue to actually see changes these days in our forests, right? The globe is changing in a variety of ways, and one of those ways is that our forests are starting to change as well. So in this graph here, what we're looking at is each of the red points is showing an area that is changing from an ectomycorrhizal dominated system to an arbuscular mycorrhizal dominated system. So we're seeing a lot of transition, especially in our eastern forests. And understanding what drives and sustains this pattern is important to our understanding and predictions of future forest structure, as well as the biogeochemical consequences, such as carbon cycling. So if mycorrhizal fungi contribute to these large-scale patterns that I've described here, then what local scale factors select for one mycorrhizal guild over the other? Or in other words, what is happening at the root zone that could be contributing to these broader patterns? Put a little more broadly, how do these factors then relate to our understanding of current forest structure? And how does this influence our predictions of the effect of global change on forested systems? However, mycorrhizal comparative studies are challenging for a number of reasons. But one of those reasons is that most tree species associate exclusively with members from one of these two mycorrhizal guilds following their seedling development or once they're mature. So therefore, comparative studies would often involve multiple unrelated host tree species, which introduces a lot of confounding variables both above ground and below ground. So it's really hard to make comparisons between ectomycorrhizal fungi and arbuscular mycorrhizal fungi because you'd have to account for the different plant host species, and that's really challenging to do. However, there are certain tree species, such as members of the genus Populus, thinking cottonwoods, aspen, and Salix, Alnus, eucalyptus, that possess a unique ability to associate with both arbuscular and ectomycorrhizal fungi simultaneously throughout their lifespan. And we call these duly colonized species. And this makes them really useful models for AM and EM comparative studies. So since duly colonized species can theoretically exist along a gradient of primarily arbuscular mycorrhizal focused to primarily ectomycorrhizal focused, we can use them to potentially look at, on a micro scale, the patterns that are playing out on larger geographic and evolutionary scales between plant species. So we're using them sort of as a model of like, if something tips the scales in a duly colonized species to be more heavily colonized with EM fungi versus AM fungi, are those the same factors driving these large scales across latitudinal patterns? So that's what I became really interested in during my time here at the University of Wisconsin-Madison. Oh, and there's a picture. I forgot I included this. This is what a duly colonized root looks like. It might be kind of small on your screen here, but essentially you're looking at a plant root, and we're seeing that fuzzy little tube sock, the ectomycorrhizal mantle here on this little newly formed root nub. And you can sort of see in here some of those larger structures are arbuscular mycorrhizal vesicles and some of the arbuscules in there as well. So you can see both types of fungi on the same root. So my research is primarily involved around using a duly colonized populus system, both cottonwoods and aspens, which I'll talk a little bit more about. So my research is aimed to assess the influence of both abiotic factors as well as genetics, and also this carbon and nitrogen economics, which I probably won't get time to talk about, but I've left the slides at the end just in case. But sort of assessing this three-tiered system on which and how they're affecting mycorrhizal colonization. All right, so I'll highlight a little bit of what I did in chapter 1, but essentially it was my first study that I did here, where I was looking at abiotic factors, or essentially environmental factors on mycorrhizal interactions. But I talked a little bit about carbon costs, right? And how accessing nitrogen is sort of expensive to a plant. But what determines whether a plant potentially has the carbon budget to afford these costs? What determines a plant's carbon budget? One way to look at this is the photosynthetic rate, right? Photosynthesis is how much sunlight they're converting into carbon-rich substances. And one obvious factor controlling the photosynthetic rate is light availability, both in quantity and quality here. But essentially, as you have more light, you're also going to have a higher photosynthetic rate, or at least versus light limitation. And another is soil moisture. In low moisture conditions, plants actually shut down their stomata, or the little holes in their leaves, to avoid losing water via transpiration, which reduces their photosynthetic rate overall, which could lead to less carbon as well. So the central question I was chasing in the study was, how do certain environmental factors related to mycorrhizal costs and benefits affect the total and relative colonization of ecto and arbuscular mycorrhizal fungi in Populus deltoides, which is eastern cottonwood? And put sort of briefly here, I did a greenhouse experiment where I had different shading shelters. They all had different light levels of 3 different levels. Also, there were different watering frequencies. So there were different soil moisture metrics. And they were given different nutrient sources as well. So we had 2 nutrient addition treatments where they either received a mineral nutrient, sort of a freely available NPK fertilizer, like what one might put in their yard or houseplants. But also some chopped leaves. So an organic source, it's a little bit more challenging to break down. And there was also another one that was a no addition treatment where all it had was just whatever nutrients were in the background soil that was in all the pots. So we had both addition treatments where they got more nitrogen, more nutrients added and in different forms, and then 2 that received, overall an organic source, something that would require enzymatic capabilities to potentially break down the nutrients. And so I'll highlight just a little bit of what we found here due to time. But overall, what we found is that Populus deltoides, eastern cottonwoods, can shift their mycorrhizal investment strategies in adaptive ways. So let me put that, I cut to the chase a little too soon here. So essentially across all environments. So here, the size of the point we're looking at is the light treatment. The color here is the different nitrogen source. And the shape here is the different moisture level. So across all of these environments, there was a strong negative correlation between AM and EM colonization, which is evidence for a biological trade-off between these 2 guilds. So in other words, when a plant has higher colonization in one guild, such as over on this end where we have higher AMF, we tend to have lower EM colonization. So you can see that depicted here. So as you get increased EM colonization along this axis, we have reduced AM colonization. So these plants are facing some kind of a trade-off. I'm going to have more of one and less of the other, or more of this one and less of this one. And this trade-off was driven by both nutrients and lights. So we're looking here at color as well as the shape of the point. And I have a few little circles here to help us out. So in low light and inorganic, the NPK fertilizer treatments, these are both clustered at the high AM, low EM investment end of this trade-off. And that sort of makes sense because if you have freely available nutrients, perhaps you're not really interested in the enzymatic mining capabilities of EM fungi, but you also are on a pretty low budget as well. You have low moisture and low light. So we would suspect these points to hold down this end of the trade-off. And on the other hand here, the no addition, where you have not only a lower amount of nutrients, but also they're of a really tough quality to be able to break down. So you perhaps would want to associate, or you would benefit from associating with EM fungi given your nutrient constraint. However, in high light and high moisture, where you're not limited by carbon, those points tended to associate with a lot of EM fungi and not as much AM fungi. So they tended to be on this end of the gradient. So overall, what this indicates is that Populus deltoides can shift its investment strategy in adaptive ways across these different range of environments related to costs and benefits. So additionally, one thing you might notice, because the colors are like the most prominent thing on here, is that you may have noticed that all of these kind of like Dijon mustard colored points are located at this end. So the other ones you can kind of see, oh, we have some purple sort of spanning here and the green sort of spans, but these mustard colored points, the ones that didn't receive as many nutrients, they're all clustered at this end of the gradient. And this suggests that when nutrients are both limited in quantity and quality, so they're not only not very accessible because they're not around, but they're also really challenging to access, plants are seemingly restricted to the ectomycorrhizal nutrient acquisition strategy. And that's regardless of other environmental factors at play here. So nutrient status was therefore the primary factor influencing this trade-off. All right, so that was a little abbreviated snapshot of what was going on in chapter 1 there. In chapter 2, this was a field study where we were looking at the role of genetics, and this was a collaboration project with the Lindroth Lab at the University of Wisconsin-Madison Department of Entomology. So in chapter 1, we looked a little bit of like what different environmental factors are driving it. And in this study, we're looking a little more like plant centric in terms of how much is genetics also controlling this. So to test the effects of genetics, we intentionally removed variation as much as we could in the environment using a common garden. And so a common garden is essentially a big garden in this case that has replicates of different genetic clones or different genotypes that are spaced in ways that you can sort of account for environmental variation, and they're all in the same place. So it's a system that's often used to test genetic questions. So what we did was we collected roots from each tree that were traced back, so we knew exactly which genotype, exactly which sort of genetic clone we had. We also collected live leaves and abscised leaves, or leaves that were falling off of the trees, and we made sure that those were from the exact same tree too. And we took soil samples at the same time that roots were collected. So we did a bunch of chemical assays on all of those tissues, and we also did microscopic quantification of both AM and EM fungi, but there wasn't very much AM, so this became very much an EM, an ectomycorrhizal project. And what we found was that ectomycorrhizal colonization was explained by genotype, or sort of by genetic individual and clone. And it had what's called a broad sense heritability of 0.26. In other words, what that means is, so broad sense heritability scores are between 0 and 1. And so essentially what this means is about a quarter of the variation in ectomycorrhizal colonization was due to genetics, which is actually a pretty high score. So I didn't show it in here, but the chemistry for the abscised leaves had a broad sense heritability of about 0.33. And we tend to think of like actual plant traits like leaf chemistry or something as having high heritability, being something that is highly genetically encoded. But we don't always tend to think about the degree of ectomycorrhizal colonization as being something that's strongly genetically encoded, but that's exactly what this study did show. We also found that EM colonization was positively correlated with the carbon to nitrogen ratio of abscised leaves. So abscised leaves are the ones that they're right before or as they're about to fall. So the plants have sucked up any nutrients that they're acquiring. Think of in the fall in the Midwest when the trees are changing color, they're going through a unique process where they're soaking up as much energy and nutrients as they can from those leaves. But what we looked at here was the carbon to nitrogen ratio, which is oftentimes used as a metric of how easily decomposable these leaves are. I've included a little figure here at the bottom. So as that ratio increases, the decomposability of those leaves goes down. So we're looking essentially along an axis of recalcitrance. How easy are those leaves able to be broken down? And as the recalcitrance increases, so does the degree of EM colonization, which is sort of how we'd predict, given what we saw earlier, is that if they're harder to break down, you may invest in more EM fungi to be able to access nutrients from those leaves. And this is not only just at the phenotypic level where we look at each tree, but also when we summarize these based on the actual different genotypes, when we average out each clone of each genotype, or what we see here in the red points, we see it's the exact same trend too. So it shows that this is sort of a genetically coded thing and has been co-selected potentially over time. Additionally, and also pretty interestingly, we found that ectomycorrhizal community composition, so which species were there in the roots, also diverged as this C to N ratio or decomposability of leaf litter increased. So again, we're looking at this C to N ratio of abscised leaves or leaf litter. And as that increased, or as the leaves became less decomposable, harder to break down, we saw that the community diverged and shifted. But most notably here, these little symbols are hard to see, but they're like a peroxidase enzyme icon. What we can see here is that Quaternarius, which some of you may be familiar with that genus already, just being on forays and things of that sort as well, it was our most abundant taxa across all of the samples that we sampled. And it's well known for its strong enzymatic capability through class 2 peroxidases. So we see that as these leaves became harder to break down, they not only were investing in more ectomycorrhizal colonization, but also different ectomycorrhizal fungi. And most notably, we see a strong increase in Quaternarius. So pretty interesting. And when we look sort of across all of the EM fungi that were present in these roots as well, and we sort of went through and we said, okay, do they have documented enzymatic capabilities, broadly speaking. It could be anything, peroxidases, lactases, Fenton chemistry, anything that is sort of known to be able to break down plant cells and access nutrients. We actually see that those that have those capabilities tend to have more positive relationships with the abscised leaf decomposability than those without them. So I wanted to see what time we were at here before I jumped into chapter 3, because it's a little bit of a long one. So maybe I'll leave a little extra time for questions on that. But thanks for letting me share a little highlight from some of the work that I've been busy with over the last many years.
Wow, thank you so much. Do people have some questions? I'm still sort of digesting a lot of it. I think what I found was really fascinating was the studying of the poplar trees, or the clonal things, and hearing that it was genetic versus, like, if I think about, like, when you're in Colorado, right, and you see like swaths of poplar that go for like miles, and
Totally.
it's possibly clones, and it's like, are there some areas where the light and the soil and like everything's different, but like maybe genetically they've already kind of made their choice, right? Like, this is what we do to survive, versus can they adjust, and then does that change their offspring because now it's changed them genetically? Does that make sense?
Yeah, I think I'm following. And actually, I was looking here because I hid this slide. There's a lot in here, but I think it'll show it. Oh, it probably has some animations here that I can show you. Yeah, so I mean, you pointed out a great thing, Kathy, is that, like, if you're familiar with aspens in particular, they have this life history strategy of living in these clonal stands, right? And that's actually one of the things that made them a perfect system as well for this study, is that if in any tree, perhaps if we were to see ectomycorrhizal colonization, or even the species that they were associating going to genetic fixation, we might see it in ones that have a life history of clonal stands. So in this slide that I show here, I show a tree, and imagine this looks more like an aspen than it does here. But in one case, they persist in these clonal stands where perhaps over many generations, evolutionary time, they're getting very consistent litter inputs. And litter inputs, I mean, like, leaves in particular. And it has essentially the same chemistry or small variations, right? You might see that those trees, become really efficient at having just the right amount of ectomycorrhizal colonization, or associating with, just the right community to be able to, like, very efficiently break those down. And you have a really tight feedback loop of nutrients in leaves, nutrients into ground, nutrient acquisition strategies here, and it's a very tight loop. However, even within aspen clones and things, there's all kinds of nutrient inputs. And here I'm highlighting mostly just potentially different trees that are in the stand, around the stand. Leaf litter blows around, and there's all kinds of stuff. Ecology is messy, right? But perhaps if there have been different mechanisms over time as well, and it's not as tight as I'm showing in this picture on the left here, you get these varied nutrient sources, and perhaps that degree of feedback is pretty loose, and actually it's more just a response to the environmental conditions of what's there at the given time. We're going to respond to that, versus having something that's been co-selected and part of what people call these adaptive trait syndromes, if you will, where these sort of suite of characters that have been finely tuned for a particular life history or an environment have been genetically coded and paired. It could be one or the other, or it could be both, right? This is kind of like a really intense dichotomy. It's likely something in the middle. That's kind of what we see. But the fact that we did find that ectomycorrhizal, the amount of colonization was, had a heritability of 0.26, that's like a quarter of the variation. That's a pretty high value, which is kind of cool.
Yeah, that struck me the most, when you kind of, for us, because we're not so deep in it, the numbers don't mean a lot, but for you to say the normal is this, so this is a lot.
Yeah.
They actually, it essentially chose a strategy.
Yeah. I think the first time I did that calculation too, I was like, okay, 0.2, like, I don't know what this totally means. And then it was, as I'm looking at it in comparison to the other plant traits as well, I was like, oh, this is just as high as, live leaf chemistry, upsized leaf, like plant chemistry values. It's basically the same level of heritability, which is pretty cool.
Wow. So any other questions? Does anyone? I thought another interesting takeaway was, oh, here's one. So Alicia Waters is asking, how might this research inform strategies to address climate change? I've often wondered about the relationship among plants and fungi and how those relationships are affected and what the repercussions might be. When we move trees into areas they haven't historically been. So with assisted migration, we had an assisted migration lecture last year. I've asked people in the field about this. Now we get blank stares.
Yeah, well, I mean, part of the blank stares might be because that is a complicated question, certainly, but a very interesting and a very, very good one. I think what sort of strikes me the most here in terms of like how to think about this or use some of this, because, even just wrapping up, I've kind of gotten to the point now where I have this information and I'm like trying to synthesize it all a little bit more too. But, it becomes really important to think about, like, first of all, like what sort of is currently that map, like this figure that I sort of showed you. I presented this as sort of a theoretical thinking of like, we think this is why this is going on, but we don't totally know all of this 100%. And we don't totally know like how resilient it is to change. Like sometimes feedback loops have cool mechanisms of like self-regulating themselves, but sometimes if you get them off course, they can start spiraling in a brand new direction. And given that we're seeing forests changing, it's possible that we're having some of that happen, whether it's just changes in temperature, which are increasing the window for decomposition. Maybe that's been a big one that's sort of restricting these areas too. We also have a lot of nitrogen deposition as well. So nitrogen is becoming more accessible. There's more time for things to be breaking, for microbes to be breaking things down. I guess, yeah, it becomes just sort of fascinating to think about like what's driving it, and then think about that in the context of like what is actually changing in our forests. If it is driven by nutrients, how are nutrients currently changing, given sort of the new way of the world, even within the last like few hundred years. And I didn't talk about the third chapter, but this might speak a little to your question as well. But we've been trying to incorporate, I didn't do this. I don't do like big models and, those kind of things, but a lot of people who do these climate change models and predictive models are incorporating mycorrhizal fungi now, which is really cool and really important. But it oftentimes, there's so much we don't know about them, and it forces us to really put a discrete number of like, okay, these interactions cost this much carbon for this much nitrogen, and it's a very fixed rate and it's very estimated. But what we did find in the third chapter is actually that rate changes depending on context. And so there's this sort of like market dynamics thing going on as well too, which makes things further complicated. But that may have been a long-winded answer to your question here. Let's see if I can actually find the chat. So.
Yeah, Alicia, did you want to unmute and ask anything else?
No, yeah, I know it's really complicated. I'm just, interested in kind of all the information that's out there, however little there may be at the moment. It's just a big bunch of questions that I have that I don't think a lot of people are even asking or considering when we talk about this in the context of climate change. So thank you.
Yeah, of course.
Yeah, I think we did had a lecture once, and it talked about, I think people were measuring like when, it was more of the saprobic things, but like something falling and rotting slowly and returning to the earth is way less of a carbon release than chopping it down and making a fire with it, basically.
Yeah, totally.
So it's like that rotting is sort of natural, and it does sort of sequester some of that carbon, which is nice. I like to see forests that have a lot of rotting stuff in them too, because I feel like there's more mushrooms.
Absolutely. Absolutely. And then you get cute mosses with them too, and you just get the whole fun team.
Perfect. Ryan is asking or saying, I saw a lot of red dots that changed from ECM to AM over the Midwest. Can anything be done to conserve ectomycorrhizal diversity in the Midwest?
Yeah, that's a good question. And there are certainly probably people who are better suited to speak to this than I am. I think, one of the factors that they think is closely tied to this too is fire suppression. So a lot of ectomycorrhizal species tend to be more fire adapted and rely on fire. So even the examples I gave at the beginning are like most coniferous species are ectomycorrhizal, oaks are ectomycorrhizal, not all, but a lot of these species tend to do better in fire. If there's a broad distinction to be made. So that could be part of it. And it could even be how nutrients are cycling with fire. So if you have fire rolling through areas, you tend to get, a quick sort of loss of nitrogen in that area in a way. From what I understand of the fire dynamics and nutrients, that is also more complicated than I'm making it sound. But you have these times where all of a sudden all of your nitrogen is volatilized. And perhaps those are really important by creating these low nutrient conditions that select then for plants that are going to be associating with ectomycorrhizal fungi. So those are kind of the things that I tend to think about in those areas is that, even up in boreal areas where you don't tend to have the regular rolling fires that you sometimes see in more temperate areas. Those areas are still sort of adapted to fire in many very important ways.
Interesting.
Cool. Thanks, Kathleen.
Yeah, thanks.
Great talk.
Likewise.
Yeah, it's so fascinating. I was going to say one more thing, just because I'm thinking when you were talking about Cortinarius kind of being at the top of like maybe providing that relationship to break down the nitrogen. And there's a place where I like to go hunt in a certain time of year. It's mostly oaks, but there's a lot of Boletus. And then in certain areas, it's all Cortinarius. And it makes me look differently at that place. Like, it's like, I think of it as such a lush woods, but it's nearly all oak and it's very rocky and it's on a cliff kind of thing.
Totally.
So like, oh, they're working really hard for their nitrogen to have like, kind of partner with all of those Cortinarias.
Absolutely. And I think it's cool too when you think about it of like, well, I guess that's how you framed it too, is like the Cortinarius is working hard, but the plants are also working hard to afford those type of more expensive symbionts as well too. So it's cool to like synthesize some of that too. A lot of this work is from the plant perspective in a way. But it's interesting to sort of put it all together and try and think of it as like what ecology looks like in a coherent way of these dynamics of interaction, I guess. I don't know. Yeah, it's really fun.
Very cool. Wow. All right. Any last questions? Anyone want to share anything before we go enjoy the last little bit of the weather today? Let's see. Let's see. John and Claudette, this is just another example of our environmental change or evolutionary change. But most of the morels we're finding to our forays were associated with cottonwoods rather than the classic dead elms. Stumps still there, but seems to be shifting as stands of classic dead elms become fewer. Yeah, I wondered that too. I've found morels in places where you would never think you'd find them. And I wonder if the trees are evolving or the mushrooms are evolving or both to kind of find their hosts and work together. That's a really great point, John and Claudette. Yeah, it makes you, it just sort of like you walk into the woods and it's like, oh, it's a beautiful woods, but just learning this info makes you look at things differently. So thank you for that.
Of course. Yeah. And, the pairing between morels and cottonwoods I think this is the first year too. I've heard people mention years previously of finding them near cottonwoods, and morels are just so dynamic. It's hard to even know what's going on with those. But now that I've spent years studying cottonwoods, I feel like cottonwoods are a pretty dynamic host species. And so in some way I'm like, oh yeah, they seem to go hand in hand in my brain now of just being like, they're doing a little bit of this and a little bit of that. And we don't quite know what they're up to, but they're just like living in these kind of in-between spaces. So
Cool. Awesome. Oh, well, thank you so much for joining us tonight, and congratulations on your PhD. It's just really impressive, and we're honored to have you here talking to us tonight. And same with you, Ryan. Honored to have you being such a longtime member in our club and coming back and presenting your research. So thank you all.